Thanks for inviting me to be part of this, Ben. This is really a neat initiative. I'm going to start by introducing a journal that FEMS has just launched that I'm heavily involved with, and this is called FEMS Microbes. And our tagline for this journal is Open Science Toward a Sustainable World. And the reason for this tagline is because we are really interested in publishing manuscripts covering all of the microbial sciences. And so our scope is to publish both basic and applied microbiology papers across the areas, so including industrial microbiology, environmental microbiology, clinical and veterinary microbiology. And we're dividing our journal up into a couple of different areas. And so we hope that this will really cover the spectrum of microbial sciences. And you can see where your submissions could fit into the journal. And so these include microbes and metabolism and multicellular life, disease, environment, and microbes and society. In addition to these sections, we publish a number of different kinds of manuscripts. So we have research articles, of course. We're also publishing short review articles as well as commentaries. And so we invite pre-submission inquiries, particularly for those reviews and commentaries. This journal launched a few months ago, and our first published articles should be going online next week. So we're really excited about that. And we think that this journal has a couple of interesting properties that we want to point out to you. And so first, these are the folks that are involved in the journal. And so you can see on the right, the editors-in-chief are myself, Jenna Jass, and Kathleen Scott. And so it doesn't take a lot of attention to realize that we have an all-woman editorial board, and this is really exciting to us. This wasn't intentional, it just happened this way, but many editorial boards do not have this gender makeup, so we're kind of excited about this. More interesting is the diversity in terms of geography that our editorial board represents, and so we have covered Europe, Southeast Asia with me, as well as North America. And our handling editors, the rest of our editorial board that you can see on the left, also is a very diverse group of people covering a range of microbial sciences, a lot of different geographical sites, different career stages as well. And so this is something that's also a really important aspect of the journal for us. So some details about our journal that might be interesting for you to know about. So we have what we consider progressive publishing policies. So we are very open to and actually encouraging of people to publish or post their work on preprint servers prior to publication. So we encourage this. We have a very transparent peer review process as well. So authors have the option to publish all of their peer reviews as well as the author's responses to those reviews along with their manuscript at the time of publication. In addition to that, the peer reviewers themselves have the option to have their names included with their reviews. And so there's the option of full transparency if everyone is interested as well. We also, in the interest of transparency, recognize co-reviewers. We appreciate that many reviewers invite trainees, postdocs, PhD students to help with the reviews, and we think this is very valuable. But we recognize that those co-reviewers don't always get credit for their contributions, and so we will be recognizing those co-reviewers, and they can get credit via Publons for their contributions to the reviews. We offer scooping protection, so if a publication comes out showing what you've demonstrated while your paper's in the pipeline with our journal, that has no negative consequences for you at all. We offer discounts for FEMS-affiliated societies, for publication fees, and our publisher, Oxford University Press, as well as FEMS, the society, do a great deal of work to help promote our papers. And so what that means for you is that if you publish your paper in FEMS Microbes, it will experience increased visibility because of this behind the scenes work from everyone involved. And then finally, one of the, I think, most important things from my perspective about this journal is our strong dedication to incorporating early career researchers at all stages of the journal. So we are actively building a pool of early career peer reviewers. This has been sprinkled around social media as well, and FEMS will be formally inviting people to submit their names to our growing database of early career researchers for peer review. We are calling for submissions for thematic issues, and we'll particularly pay attention to those submissions coming from folks that include early career researchers as part of the guest editorial team for that thematic issue. FEMS hosts a number of webinars where we highlight papers that we're publishing, and so this is a great opportunity for early career researchers to get their work and their faces out there to the public and share their research with the world. And then FEMS is also really active on social media, and so there's a number of ways to get involved with that as well, as you can see here on this slide. So if you have more questions about FEMS, there's a lot of information online, and so you can check that out at these different links. You can drop me a question if you're listening live into the chat here. And we look forward to hearing from you and seeing your submissions down the road. And so with that, then I'm going to switch and tell you a little bit about the research we've been doing here in my lab in Singapore, where we focus primarily on the bacterium called Enterococcus faecalis, and we are most interested in its ability to cause biofilm-associated infections. And so we're motivated to study this organism because it is always on the serious threat lists that are put out by, for instance, the CDC or the WHO for its high prevalence of antimicrobial resistance. And so you can see the numbers here on this slide. It's often associated with resistance to antibiotics of last resort. So this is a major clinical and public health problem. So there's two species of Enterococcus that most frequently cause infection in humans, and those are E. faecalis and E. faecium. We study primarily E. faecalis, and this is a commensal in the GI tract of probably everybody listening to this talk. It's just part of the healthy human gut microbiome. However, when things go awry in susceptible hosts, it can get out of the GI tract into other sites and cause opportunistic infections. And so it's most frequently associated with catheter-associated urinary tract infections. In people that are exposed to broad-spectrum antibiotics, it can grow out of control in the GI tract, and this can lead to infections in other sites, such as the bloodstream. And it's also associated with a wide variety of wound infections, including diabetic foot ulcers. And so in my lab overall, we're then interested in understanding how E. faecalis perceives these different host environments it finds itself in, and how it responds accordingly by, for instance, the elaboration of surface structures that allow it to thrive in these environments and to also form the biofilms that we know are really important for all of these different infections. And so today I'm going to tell you just a little bit about the work that we've been doing in the context of wound infections. So I just mentioned that biofilms are important for these wound infections, but it's not just E. faecalis biofilms. And most of these infections, and actually most of the infections it causes across the body, these are not monomicrobial biofilms. These are often polymicrobial interactions that E. faecalis finds itself participating in. And so this is something else that we're really interested in understanding. How does E. faecalis interact with other organisms at these infection sites? And so I'll tell you again a little bit about the work we've been doing in the context of wound infections. So we use a mouse model of wound infection in the lab, and what we see is that if we infect these mice with about 10 to the 6th bacteria, within a few hours after infection, we see this very acute and rapid replication of the bacteria within the wound. However, as time progresses, what we see is that the bacteria sort of level off at this chronic persistent infection level. And we think this is important because this reflects what happens in humans experiencing chronic wound infections, where the bacteria just are not cleared well by the human immune system. They just hang out for long periods of time. There's some other features of this wound infection model that also reflect what are seen in human wound infections. So these are biofilm-associated infections, and we can see readily these little microcolonies of E. faecalis scattered across the wound bed. And in addition, in humans, wound infections are associated with delayed healing, and we also see this in the animals. So here on the bottom, this is a cross-section of a wound that was not infected. 7 days later, what you're looking at here is a completely healed wound. Whereas on the top, this is an infected, E. faecalis infected wound 7 days later. This is not healed. You can still see a very clear scab. And in the underlying tissue, there's no clear signs of healing yet. So this is clearly delayed wound healing. So we wanted to understand then how E. faecalis was causing these outcomes within the wound infection. And so to start this out, we looked at the immune response at these different stages of infection. And so during this acute replication period by E. faecalis, we looked at the cytokines and the chemokines that we could observe at the site of infection. And so what you're looking at here in this spider plot is animals that are mock infected in black, and then the animals that are infected with E. faecalis in blue. And what you can capture here is that there's a strong pro-inflammatory pattern of cytokines and chemokines being produced during this acute infection. However, if we look at these later infection stages during this persistent infection, the picture is quite different. So you can see that the blue and black lines overlap predominantly, so there's not a strong immune response. And if anything, some of these blue lines, these infected animals, dip down below the mock-infected animals. And so this started to hint at us that there is some degree of immune suppression happening during this persistent infection. And so we can also see that in the immune cells that respond to the infection site. We can look at neutrophils, and again, during acute infection, we see a strong neutrophil response, which is what we would expect and is consistent with the cytokines and chemokines we observed. And this dissipates over time. And if we look at macrophages, it looks the opposite. We see very few macrophages early in infection, and this starts to go up over time. And so this presented us with a conundrum. So if these immune cells keep coming in during the chronic infection, why aren't they clearing it? And so to address that, we turned to some in vitro models to look at this. So here you're looking at macrophages in vitro. We use an NF-kappa B reporter assay to look at the degree of macrophage activation. And we asked the question of whether E. faecalis could actually suppress immune responses in this experiment. So we activate our macrophages with LPS, which will maximally activate them. And then we add increasing numbers of E. faecalis to the cells. And what we see is a really strong dose-dependent pattern of immune suppression of LPS, suppression of LPS activation with E. faecalis. And this is in the absence of any cell cytotoxicity. And so LPS is kind of an artificial way to do the experiment. So we also asked if we could see the same thing with a co-infecting organism. And so here we infect our macrophages instead with E. coli. This also by itself can activate the macrophages. And again, E. faecalis can actively suppress that E. coli-driven macrophage activation. And so this then became pretty interesting to us because I told you E. faecalis often participates in these polymicrobial infections. And so we wondered if this might be important for these mixed species infections. And this interaction between E. coli and Enterococcus is relevant to real infections. This is metadata from bacteria that are most frequently cultured from diabetic wound infections. And so here you can see Enterococci are in the top 3, along with Pseudomonas and staph, but E. coli and Klebsiella round out the top 5. So we wanted to then understand even better this interaction between E. coli and Enterococcus within wounds. And so now we go back to that wound infection model I just showed you. If we infect E. coli by itself, we see that the animals have relatively low titer E. coli wound infections. Each of these dots is the number of bacteria in one mouse. And so we see 100 to 1,000 bacteria per animal. But if we co-infect with E. faecalis, this changes dramatically. We see many of the animals have extremely high titer E. coli wound infections. And so we wanted to understand this better. What is driving this high titer wound infection? And so we can model this, what we see in animals, also in the laboratory, in vitro. We can grow biofilms in different assays and look at these interactions. This is a macrocolony biofilm assay. And these macrocolonies are 5 days old. So this is E. coli by itself, E. faecalis by itself. And if we start to mix bacteria together, at a 1-to-1 ratio, we don't see very much. But at the time of inoculation, if we increase the ratios of E. faecalis to E. coli, so there's a little more E. coli or E. faecalis in the mix, we start to see these really dramatic changes in colony morphologies over time. And even though there's more E. faecalis at the start of this experiment, this outgrowth is actually due to E. coli. And we know that we can see that a little more clearly here in these proximity assays, where on the right we spot E. faecalis along this diagonal, and on the left we spot E. coli. And what we can see is that the E. coli grows into much larger colonies the closer it is to E. faecalis, and we can quantify that as well. So these E. coli are really proliferating as they get closer to E. faecalis. And so then to understand how that was working, we did some transcriptional expression profiling within E. coli close or far away from E. faecalis. And we find that the genes that are most highly upregulated in E. coli are those involved in iron acquisition, and specifically in the production of siderophores. And so what siderophores are these small molecules that are made by E. coli and exported into the extracellular space. They bind iron with very, very high affinity, and then the E. coli can take up those iron-bound siderophores, and this serves as a source of iron. This is important because iron is an essential nutrient for E. coli growth, and iron is often very limited in the environment, particularly during infection. And so we have mutants then in each of these siderophore biosynthetic pathways in E. coli. There's 3 of them, Enterobactin, Salmonellin, and Yersinia bactin. And so we then asked, do those mutants grow to bigger colonies just like the wild type E. coli does? And I think you can see in this picture, the answer is no. In panels A and D, these are Enterobactin mutants. So if E. coli doesn't make Enterobactin, it cannot grow better in proximity to E. faecalis. So this is what we see in vitro, but we're really interested in what's happening in that wound infection model. So we now step back to the animals and ask if the same thing is true. And it turns out the answer is yes. So I told you that if we co-infect wild type E. coli with wild type E. faecalis, we see that most of the mice have very high titer E. coli wound infections. But if we instead co-infect with that Enterobactin E. coli mutant, this is not able to get the benefit from E. faecalis and grow to higher numbers. So Enterobactin is important for that enhanced E. coli growth in the proximity of E. faecalis. So how does E. faecalis drive this in E. coli? And so through some genetic screens that I don't have time to tell you about, we discovered that one particular gene, ARCD, which encodes an antiporter called ARCD, seems to be important for driving that interaction. So ARCD simultaneously imports arginine and exports ornithine out of E. faecalis. And so we hypothesized that that exported ornithine might be serving as a cue for E. coli to grow better in proximity to feces. So to test that, we add ornithine directly to E. coli in the absence of E. faecalis, and we see indeed ornithine can drive E. coli biofilm growth by itself. So it's sufficient for that enhanced biofilm growth. And it grows biofilms then as well as E. faecalis, the E. faecalis mix does. And really importantly, this is dependent on Enterobactin, because if E. coli doesn't make Enterobactin, ornithine has no effect on it. So then together, all of this says, in vitro, E. faecalis exports ornithine. This serves as a cue for E. coli to grow better in an Enterobactin-dependent manner. And so lastly, then we go back to our animals and ask, is ARCD and ornithine also important in vivo? And here the answer is a little bit mixed. And so again, E. coli by itself it has really low titer wound infections. The mixed species wound infection, we have lots of E. coli in the wounds. If we co-infect with wild type E. coli and the ARCD mutant that doesn't make ornithine, we get an intermediate phenotype. We have fewer animals with high titer wound infections, but we still have these animals, a residual subset of animals with very high titer E. coli wound infections. And so this then led us to hypothesize that the immune suppression that I told you about at the beginning of my talk might be responsible for this residual growth of E. coli. And that turns out to be the case. We now have mutants in E. faecalis that are unable to suppress macrophage activation. They can't suppress the immune response. And so if we instead co-infect E. coli with those E. faecalis, so there's a stronger immune response essentially, we lose those residual high titer E. coli wound infected animals. So what this says together is that E. faecalis has a couple of mechanisms that are ultimately beneficial to a co-infecting organism, in this case, E. coli. For one, it helps E. coli acquire iron by this constitutive export of ornithine, leading to Enterobactin production and iron acquisition. And simultaneously, by a completely different mechanism, E. faecalis is suppressing the host immune response. And we know that E. coli by itself is very pro-inflammatory. So this, we think, has an added benefit for E. coli as well. So taken together, we have started to think of E. faecalis as kind of a Jekyll and Hyde organism. So in normal healthy hosts, if it stays in the GI tract, it's a completely innocuous and often helpful organism. but due to its ability to both modulate the immune system and participate in these interesting polymicrobial interactions, it can turn into a bad guy. And so we call that Mr. Hyde. And so, of course, we're really interested in understanding then how faecalis interacts with other organisms that it finds itself with within different infection sites. And so that's some of our ongoing work. And so with that, I will just thank very much the people whose work I showed you today. This is Kelvin, this is Brenda, Patrick, Wei Hong, and Damien. And I will stop there and thank Ben again for the invitation to join you today.
Thank you very much, Kimberly. Fascinating stuff, both with the journal and this really cool and complete story. Unfortunately, we don't have time for questions here, but is there anywhere people can kind of ask you stuff and get a hold of you?
Yes. Let me just go back to my first slide. I have my, hold on. Let me do it this way. Here we go. Kimberly at Kim in Singapore is how you can find me on Twitter, or you can go to my lab website and you can find my email address there too.
Perfect. Thank you. I'm sure we'll get lots of questions. I look forward to hearing more about your research and more about the journal. Thank you very much for your time and your talk.